Hello everyone. Today my presentation topic is on the DCOG Plus project, which is focused on the identification of dietary modulators of cognitive function in aging using metabolomics discovery. Today I'll be focusing on the discovery phase of the project, and this is a partner collaboration between 6 different universities. So I'm going to start off with a brief introduction of neurogenesis in this context. First, we believe that dietary components can modulate neurogenesis and cognitive function by communicating with the systemic environment around the blood vessels in our brain. Throughout our human lifespan, neurons in the adult brain are constantly dying, but in fact, neurogenesis also occurs daily in the hippocampus and the lateral ventricles under normal physiological conditions. There's approximately 700 new neurons and microglia produced in the hippocampus daily, but unfortunately, this rate of new neurons growth decreases as we age. And there are two mouse studies on the right, and they show that there are certain blood circulating factors in the systemic blood value that can actually modulate neurogenesis either in a positive or a negative way. And in the mouse study on the bottom right, the results show that when a young mouse was actually exposed to plasma from an aged mouse, these young mouse experienced reduced neurogenesis as well as impaired cognitive decline. So this is a really interesting area of research. If you'd like to know more about neurogenesis, have a look at Sandrine's TED Talk. The link is at the bottom of the slide. And Sandrine is the project coordinator for our DCOP Plus project. These recent studies, including cohort studies and control intervention trials, show that certain food compounds and dietary patterns can modulate age-related cognitive function with a focus on polyphenols and the Mediterranean diet. So the Mediterranean diet is mostly characterized by the high consumption of plant-based foods which are rich in phytochemicals and monounsaturated fatty acids that are usually derived from olive oil, as well as a moderate intake of fish and alcohol and a low consumption of meat and dairy products. So as part of the discovery phase of the DCOP Plus project, we hypothesized that there are bioactive compounds from healthy diets with the ability to modulate neuronal function and delay the rate of cognitive decline. So we aim to identify a combination of diet-derived compounds that are associated with either better or worse brain aging in an elderly cohort using untargeted metabolomics with a high-resolution mass spectrometry approach. So in this project, we leverage the French 3-CD cohort study consisting of both male and female elderly participants that are aged 65 years old and above. And at the baseline of the cohort study at year 0, serum samples were collected from the male and female participants when they were all cognitively healthy, so this precedes the onset of an overall cognitive decline. And then subsequently, at 4, 7, 10, and 12-year time intervals, the participants were asked to come back for a follow-up visit for a reassessment of their cognitive function. So during this time period, some of the participants might go on to develop cognitive decline, while some might remain cognitively stable. And at the 2-year time point, food frequency questionnaires and a 24-hour dietary recall were also given to participants for a record of their food intake. So in summary, cognition was measured via a series of 5 different psychometric tests that is listed here on the slide. Cognition was measured at the beginning of the cohort study as well as each of the follow-up time points, so this resulted in multiple repeated cognitive measures throughout the cohort study for each participant, and these were eventually condensed into one single composite score to facilitate the subject selection process. In my presentation, when I'm referring to cases, I'm referring to the participants who were initially cognitively stable but then went on to develop decline either at the 4, 7, 10, or 12-year time interval. And when I'm referring to controls, these are participants who remained cognitively stable throughout the cohort study. At each of the followed time points, the worst slopes were selected as cases, and then they were matched to a control based on the same age, gender, and educational level. This is highly critical for nested case control study designs to avoid any subject selection biasness. And in summary, there were 209 cases matched to 209 controls. Now moving on to the metabolomics workflow, the serum samples that were collected at the beginning of the cohort study were analyzed with a high-resolution Q-TOF instrument in both the positive and negative ionization mode to detect a wider coverage of ions. The raw MS data was then processed using the Galaxy platform. Individual processing parameters such as peak extraction, retention time alignment, and batch correction, etc. They were all individually optimized for the positive and negative ionization mode. This is a process that has been very well established here at INRA. And in summary, there were 1,136 ions in the positive mode and 129 ions in the negative mode. In the next few slides, I will only show results from the positive mode because we are still processing the negative data. For this statistical analysis, we used a multivariate statistical approach to analyze a large number of ions simultaneously to look for any associations between them with modulating cognition. Firstly, we started off with one of the most commonly used techniques of metabolomic studies, which is the partial least squared discriminant analysis, or in short, POSDA. In this model here on the scores plot, each of the circles represent an individual participant, so the ones in green represent the controls, and the ones in the red circles, they represent the cases. So in the scores plot, we can see quite a good discrimination between the cases and the controls. And there were 147 ions that were more important in this discrimination between the cases and the controls. On the right in the loadings plot, 40 of these ions, they were more associated with negatively modulating cognition, so they are associated with cognitive decline and the intensities were much higher in the cases, whereas the remaining 107 ions, they were more associated with positively modulating cognition, so the intensities were much higher in the controls. This is quite a well-validated method, although it's not as ideal as what we would see in control intervention studies, but this is expected because we are working with a cohort study and differences that are present tend to be quite subtle. They can sometimes also be completely hidden by another factor, for instance, gender. We also wanted to control for the paired couple design as well as confounding factors. So we had to further refine the statistical strategy by having a discussion with the epidemiologist at Bordeaux, who we are collaborating with. We found that the sparse conditional logistic regression was a far more suitable method to take into account the unique case control study design as well as the confounders such as BMI, season of blood draw, as well as the number of medications that were regularly consumed by the participants. So to put it simply, this logistic regression was ran on the original dataset, and then we further enhanced this analysis by applying the bootstrap resampling, where 1,000 more bootstrap analysis was ran, and each time on a slightly different variation of the original dataset. From the regression analysis ran on the original dataset, there were 9 ions picked out as highly associated with modulating cognition both positively and negatively, and from the bootstrap resampling, a further 8 ions were selected because they frequently occurred in more than 40% of the bootstrap analysis. So in total, there were 17 ions that were associated with cognition, and from these, we screened them for potentially early endogenous biomarkers of cognition, as well as the food metabolome, which is part of the dietary intake of the participants. We are currently at the beginning of the identification phase, which is a really complex and multi-step process. It is still very much the bottleneck of many food metabolomic studies. But to put it simply, we first screen all of the known dietary biomarkers of food intake that's available in the literature, And then we annotate these ions using databases as well as commercial standards. And next, we are working with the University of Barcelona on the MSMS analysis. So this process is very much still ongoing. We have tentatively identified one of these ions, but there are 16 more ions to confirm in this positive ionization mode. Based on the first step where we first screened the known dietary biomarkers, we identified one of the most important ions that is associated with positively modulating cognition, and we hypothesized this to be atraclogenin glucuronide based on this previous cohort study. This cohort study also found atraclogenin glucuronide in coffee, and that it was a much more stronger biomarker of coffee intake in comparison to caffeine, which is expected since we can also find caffeine in tea, Coca-Cola, and even chocolates. This is the initial interpretation of the Q12 spectral data. And I just want to point your attention on the right, where you can see this ion was detected in much higher intensities in the control group, where the participants were cognitively stable. So initially we faced some challenges in obtaining atraclogenin as a pure or isolated standard because it was just not available commercially. So good thing we came across this paper where the authors were also working with atraclogenin, and we have since been in contact with them. So they have very kindly offered us a small quantity of atraclogenin to work with to compare against our ion of interest. So to everyone out there in the audience, if you do have any atraclogenin sitting in your bench tops or in your fridge or freezers, or if you're interested in collaborating to synthesize or isolate this compound, please hit me up. This is my email address here. It would be really awesome if atraclogenin can be made more widely available because it has been previously found as a strong biomarker of coffee intake in a cohort study, and also because a lot of us drink coffee. So there's a couple of previous studies showing that the consumption of coffee is associated with reduced cognitive decline. Currently, we do not know if this is due to the bioactivity of atraclogenin, but we intend to test this in work packages 2 and 3. So work package 2 is focused on testing for the ability of these compounds to modulate neurogenesis and microglial function in an in vitro parabiosis assay. And in work package 3, it is focused on testing for the protective effects of these compounds in an early life stress mouse model. So in summary, neurogenesis occurs daily, and we believe this can be modulated by diet. Currently, our results have shown that there is a good discrimination between the serum metabolic profiles between the controls and the cases, and so far there has been 17 ions picked out in the positive ionization mode. We also found atraclogenin as a strong biomarker of coffee intake, which is associated with modulating cognitive function in a positive direction. The next step, we intend to identify all of these ions that are associated with modulating cognition. So this includes the 17 ions from the positive mode, ions from the negative mode, as well as the known biomarkers of the associated foods. So for example, in coffee, besides atraclogenin, we'll also test for the other known biomarkers such as caffeine and triglyceride, for example. Next, we also intend to validate these ions in a separate human cohort study, and then to test for these ions in the in vitro parabiosis assay and in the mouse model. I've come to the end of my presentation, and I would like very much to thank our colleagues at INRA, as well as our collaborators within the DCOT Plus project. I also like to thank the JPI for funding the DCOT Plus project, and to the Air Green Skills Committee for funding the incoming fellowship to funds. Thank you very much for your attention.